So I've discussed how enhancers produce basic on-off patterns of gene expression in the early Drosophila embryo. And now I'd like to address the question of transcriptional precision. How, for example, do you get the even skip stripes turned on in the right place at the right time in embryo after embryo in large populations of embryos? And to introduce the underlying mechanisms for precision and robustness, getting that pattern right every time in every embryo, I'd like to introduce dorsal ventral patterning. That is the patterning of the back belly axis of the developing embryo, and it is controlled by a sequence-specific transcription factor called dorsal. Here you see a side view of a 2-hour Drosophila embryo stained with an antibody against the dorsal protein. And you can see that the protein is distributed in a broad nuclear gradient with peak levels present in the bottom or ventral regions of the embryo, the future belly of the fly, and lower levels in more lateral and dorsal regions, the future back of the fly. Now, this dorsal nuclear gradient controls dorsal ventral patterning by regulating something like 50 target genes in a concentration-dependent fashion. And some examples of these targets are shown on the next slide. So here's a side view of an embryo that's just 1 hour older than the one I showed you on the previous slide. This is a 3-hour embryo. And it was stained to show the expression of 4 different target genes that are directly regulated by the dorsal gradient. The gene visualized in yellow is activated by peak levels of the dorsal gradient. Those visualized in blue, green, and red by progressively lower levels of the gradient. And one point I want to make concerns that gene expression pattern seen in yellow. This corresponds to a gene called snail, which I mentioned in the previous slide. Snail works as a localized repressor to establish the boundaries of the developing mesoderm. So those yellow expressing cells will invaginate during gastrulation to form the internal mesoderm. Snail works as a repressor to delineate a boundary between the mesoderm in yellow and the lateral ectoderm in blue and green. Okay, and I'll come back to that point of snail as a boundary repressor a little later. The next slide summarizes the basic mechanisms by which the dorsal gradient produces different thresholds of gene expression. Now, if I can direct your attention to the bottom of the slide, you'll see that there's a high magnification view of the dorsal gradient, this time looking directly at the ventral midline of the early embryo. That broad dorsal nuclear gradient directly activates an immediate early target gene called twist, which is distributed in a steeper gradient. It's sharper than the input dorsal gradient. And then dorsal and twist work together to activate this target gene snail, which I just mentioned, which is sharply expressed within the ventral mesoderm with sharp lateral borders. And that snail expression pattern delineates the boundary between the future mesoderm and the lateral neurogenic ectoderm. So the dorsal gradient generates 3 basic patterns of gene expression, and these are produced in collaboration with twist and snail, which together with dorsal work as sequence-specific transcription factors. The 3 basic patterns are called type 1, type 2, and type 3. The type 1 expression pattern is represented by snail, which I've mentioned. Snail expression is restricted to the ventralmost regions of the embryo, where are the highest levels of the dorsal and twist activators. Type 2 target genes, and an example is shown in panel E, shows a narrow lateral stripe of gene expression where there are intermediate to low levels of the dorsal and twist gradients. And then finally, the type 3 pattern, seen in panel F, shows a broad lateral stripe which is activated in response to the lowest levels of the dorsal gradient. So these are the 3 basic thresholds of gene expression produced by the dorsal gradient. Now, to the right of each of these 3 expression patterns, you see summaries of the enhancers which control the expression patterns. So the type 1 expression pattern is regulated by an enhancer that contains unlinked dorsal and twist activator sites. They are uncolored, indicating that they are low affinity, suboptimal binding sites. So the only way you can turn on snail and occupy those binding sites is with the highest levels of the dorsal and twist activators. In contrast, the type 2 enhancer contains at least one pair of optimal dorsal and twist binding sites. The two binding sites are closely linked and have a specific arrangement. The asymmetric twist site points towards the dorsal site. And these features of arrangement, high affinity sites, allows intermediate to low levels of the dorsal gradient to activate the type 2 target gene. And then finally, type 3 target genes, which respond to the lowest levels of the dorsal gradient, have multiple high affinity dorsal binding sites, once again indicated by the green ball, as well as closely linked binding sites for ubiquitous transcriptional activators such as Zelda. And this linkage between Zelda and optimal dorsal binding sites ensures that the lowest levels of the dorsal gradient are able to activate the type 3 genes. You'll notice that both the type 2 and the type 3 enhancers contain binding sites for the snail repressor, and that keeps these enhancers off in the bottom of the embryo in the future mesoderm, off and off, due to the snail repressor. So altogether, these three different thresholds of gene expression in response to the dorsal gradient depend on enhancers with different binding affinities of dorsal and twist activators, with the presence or absence of snail repressor sites, and depending on the so-called grammar, the local arrangement of binding sites, such as the close linkage of dorsal and twist seen in the type 2 enhancers and the linkage of dorsal and Zelda seen in type 3 enhancers. Altogether, these features of the enhancers determine the kind of output expression pattern that are obtained in response to the dorsal gradient. Okay, so now I want to get finally to the question that I promised earlier, and that's transcriptional precision. How do you get these expression patterns on not just in one lucky embryo, but in all embryos in natural populations which are subject to all sorts of variation in genetic background and in the environment? And the first mechanism I'm going to discuss is called the shadow enhancer. And these were identified using whole genome binding assays, looking at the genome-wide distribution of dorsal, twist, and snail binding throughout the Drosophila genome in the early Drosophila embryo. And an example of these whole genome chip binding assays is shown here. So this is a dorsal target gene called SOG. It's a type 3 target gene that's activated by the lowest levels of the dorsal gradient. At the bottom here, you're seeing an embryo which is oriented so that the mesoderm is facing you. The SOG expression pattern is in the lateral neurogenic ectoderm in response to low levels of the dorsal gradient, and you see that the gene is off in the ventral mesoderm due to the snail repressor. Now, the whole genome chip binding assays identified a cluster of dorsal, twist, and snail binding sites within the first intron of the SOG transcription unit. And this corresponds to the known enhancer which regulates SOG expression. But to our surprise, these whole genome binding assays identified a second cluster of binding sites located in a remote position over 20 kilobases upstream of the SOG transcription unit on the other side of this linked gene. And despite this unexpected location, this genomic DNA fragment containing this cluster of binding sites works as an enhancer to regulate SOG expression. It produces a pattern of expression very similar to the one produced by the intronic enhancer and seen for the endogenous gene. So we have two separate enhancers separated by 25 kilobases that produce basically the same pattern of expression. Now this is different from what I discussed in the introduction regarding the Eve stripes. There you have separate enhancers for different stripes. In this case, we have two separate enhancers for the same pattern of expression. And we refer to the remote enhancer, the more distal enhancer, as the shadow enhancer. Shadow enhancers are sometimes found in really weird locations, like the introns of neighboring genes. And an example of that is shown on the next slide. So here you see another dorsal target gene called brinker, which is expressed in the lateral neurogenic ectoderm in response to low levels of the dorsal gradient. It's kept off in the ventral mesoderm by the snail repressor. The whole genome chip assays identifies a cluster of dorsal twist and snail binding sites at the 5' end of the gene, about 10 kilobases upstream of the brinker transcription start site. There's the brinker gene itself. And this 5' cluster corresponds to the known enhancer which regulates brinker expression. But again, unexpectedly, the whole genome binding assays identified a second cluster of binding sites located within the intron of this neighboring gene called ATG5, which is unrelated to brinker and is not even turned on in the early embryo. And despite this odd location, this cluster of binding sites works as an enhancer to regulate brinker expression and produce a lateral stripe pattern very similar to that seen for the endogenous gene and that generated by the normal 5' enhancer. So here we have a situation similar to what I described for SOG, two enhancers separated by about 25 kilobases, one located 5', one located 3' within a neighboring gene. These two enhancers produce very similar patterns of expression. So what's the deal? I mean, something on the order of 1/2 of the 50 dorsal target genes are thought to contain shadow enhancers. What are they doing? We certainly didn't expect them, and they certainly weren't identified using conventional gene fusion assays because of their very remote locations. And so to get at the question of what does the shadow enhancer do for development, a graduate student in the lab, Mike Perry, analyzed the regulation of the snail gene. And I mentioned before, snail is activated in response to high levels of the dorsal gradient within the ventral regions of the embryo, which will form the mesoderm. Snail is regulated by two separate enhancers, a primary enhancer and a shadow enhancer, separated by about 5 kilobases. And you see that the shadow enhancer is located within the first intron of this neighboring locus, TIM17, which again is not expressed in the early embryo, is unrelated to snail. Despite this location within the TIM17 locus, the shadow enhancer produces a pattern of expression very similar to that observed for the endogenous gene, including very sharp lateral limits of expression at the boundary between the future mesoderm and the lateral neurogenic ectoderm. So Mike Perry made a series of back transgenes which mutagenize either the proximal primary enhancer or the distal shadow enhancer, or kept both enhancers intact. And these transgenes encompass about 25 kilobases of genomic DNA, includes the entire snail and TIM17 loci and associated regulatory DNAs. In some of the experiments, Mike replaced the snail transcription unit with the yellow reporter gene. This contains a large intron which permits us to detect nascent transcripts, and as you'll see, this is the most quantitative means for assessing gene activation in the early embryo, to do in situ hybridization, detecting nascent transcripts using intronic probes. All right, so Mike made this set of 3 back transgenes, both enhancers intact, mutagenized either the shadow or the proximal primary enhancer, and an example of his analysis is shown on the next couple of slides. So here you're looking at the early activation of the yellow marker gene from these back transgenes. And we're looking at the optimal culturing condition of 22 degrees. This is the normal temperature, room temperature, that we normally raise our strains of Drosophila melanogaster. And you can see whether you have one enhancer or two enhancers, the initial activation pattern is fine. You see homogeneous expression, sharp lateral limits within the ventral mesoderm. So in this case, removal of the primary proximal enhancer does not alter the early activation in gene expression. But this is at optimal temperature, 22 degrees. At high temperatures, 30 degrees, which are stressful for the flies, but natural populations deal with it all the time, the transgenes containing a single enhancer begin to break down. and that can be seen on the next slide. So now at 30 degrees, the back transgene containing just the shadow enhancer with the proximal enhancer disrupted, this expression pattern is abnormal. You see gaps in expression, and the lateral limits start to get jagged. In contrast, the enhancer that contains, the transgene that contains both enhancers intact continues to produce a normal pattern of expression, homogeneous expression with sharp lateral limits. And so these types of experiments, and we've done many more like this, suggest that the shadow enhancer helps ensure that critical patterning genes like snail are turned on in the right place at the right time in embryo after embryo in large populations of embryos subject to both environmental variation, such as increases in temperature, and also genetic variation, which is a common feature of natural populations. So the shadow enhancer helps confer robustness. In addition, shadow enhancers help improve the precision, the clarity, the sharpness of gene expression, and I'll show one example regarding segmentation. Now, I mentioned earlier the Bitcoy gradient. It is one of the activators of Eve stripe 2. Bitcoy is already present in the unfertilized egg. The protein is distributed in a broad gradient with peak levels in anterior regions and lower levels in more posterior regions. This Bitcoy gradient controls segmentation by leading to sequential patterns of gap gene expression and, as I showed you, directly regulating pair rule stripes of segmentation gene expression, such as Eve stripe 2. The next slide shows the gap genes that are regulated by Bitcoy. This just shows 3 of the gap genes. There are several others not shown here. These are hunchback, Krupel, and Knurps, which are expressed sequentially along the anterior to posterior axis. All of these gap genes contain shadow enhancers, but I want to just focus your attention to the top panel, the hunchback expression pattern. Hunchback shows a simple on-off pattern of gene expression. It is on in the anterior half of the embryo and off in the posterior half. It is switched on in response to high and intermediate levels of the Bitcoy gradient, but low levels of Bitcoy in posterior regions of the embryo are insufficient to activate hunchback transcription. This on-off switch in transcription is controlled by a classical enhancer that was identified over 20 years ago in the labs of Nusslein Volhart and Gary Struhl. And the enhancer is located right at the proximal P2 promoter, shown in blue here, and it contains a series of optimal Bitcoy binding sites that respond to high and intermediate levels of the Bitcoy gradient to give that on-off switch. About a year ago, a graduate student in my lab, Mike Perry, identified a shadow enhancer, unexpectedly, that's located 5 kilobases upstream of the classical enhancer within the extended hunchback regulatory region, and it's indicated in red there. So this is a similar situation to what I described for snail. We have 2 separate enhancers separated by 5 kilobases that produce very similar patterns of expression. You see that the shadow enhancer gives an on-off switch in transcription very similar to that obtained with the classical proximal enhancer. So again, why 2 enhancers? In this case, it doesn't matter what temperature you look at, 2 enhancers give a sharper pattern of hunchback expression than either one alone. And some evidence for that is shown here. Once again, we're looking at a series of back transgenes that are about 20 to 25 kilobases in length and include the extended 5' hunchback regulatory sequences. The hunchback transcription unit was replaced with the yellow reporter gene, once again permitting detection of nascent transcripts using intronic probes. We have a back transgene with both enhancers intact on the right. The classical enhancer is disrupted in the middle panel, and the newly identified shadow enhancer is disrupted in the leftmost panel. And you can see that the back transgenes containing just a single enhancer give rise to kind of erratic patterns of activation. And we're looking at transcriptional activation literally within 20 minutes after the first formation of the Bitcoy gradient. So this is very early activation in response to the gradient. And you can see in the first two panels, the left and the middle panel, the patterns are erratic and the border is indistinct, and there's even ectopic expression outside the normal limits of the hunchback transcription pattern. However, with two enhancers, as seen on the right, you get a much more faithful pattern of expression, homogeneous activation, and a fairly sharp posterior border. And those numbers on top refer to the discordance between the expression pattern produced by the transgene and the endogenous hunchback expression pattern. So for example, in the middle panel, 24% of the nuclei which express the endogenous hunchback gene fail to show expression of the back transgene. So that's a very high rate of discordance, whereas when you have both enhancers intact, you only have a 3% discordance. That pattern with the two enhancers is much more faithful than the patterns produced with either the shadow enhancer or the primary proximal enhancer alone. So the shadow enhancer confers robustness. It ensures that important patterning genes like snail and hunchback are turned on in the right place and at the right time in natural populations of embryos. The shadow enhancer also improves the precision in gene activation, giving homogeneous transcription and a sharper posterior border.